Hi, my name is Brandon Shun, and I'll be talking about sensory interneurons for takeoffs in Drosophila. Halteres are a sensory organ used in flight by Drosophila in order to detect inertial forces and maintain stable flight. Information from halteres used in wing and head reflexes are sent through a direct pathway or by interneurons. Although these interneurons have been identified anatomically, their functions have not been characterized. Two genetically labeled neurons in the thorax that may receive synaptic input from halteres have been identified. Preliminary work has shown that flies with these neurons inactivated, EVAR flies, with an apoptotic transgene reaper cannot fly, similar to flies with halteres removed. The name EVAR was chosen because the gene even skipped was identified previously in the larval nervous system. Figure 2 shows these Eve proteins in larvae. The magenta is a subset of Eve plus motor neurons, and the green is a lateral cluster of Eve plus EL interneurons. The gene is also expressed in cells of adults, which is shown in figure 3. However, these animals do not show the highly variable wing amplitude exhibited by flies with halteres removed. Additionally, EVAR flies respond differently to haltere stimulation compared to wild-type flies. The goal of this project was to compare the takeoffs of wild-type and EVAR flies and quantify the role of the interneurons in flight behavior. We hypothesized that these interneurons receive information from the halteres to enable stable flight. Experiments were conducted by separating female flies and placing them one at a time on an area they could stand on. Takeoff was initiated by either vibrating the area they were standing on or presenting the fly with a looming stimulus. The takeoff and flight was recorded by using two high-speed cameras. Figure 4 shows examples of the setup as seen by the cameras. EVAR flies were created using the Reaper technique, and the procedure was repeated. A three-dimensional representation of each fly's flight path is shown in Figure 5. The starting point for each fly is at the center, and the path branches out from there. Looking at the figure, it is clear that wild-type flies have a more consistent flight pattern in the up direction, while there is no clear pattern for EVAR flies. Linearity of flight, maximum height, and velocity was compared between the wild-type and EVAR flies. Linearity of flight is the ratio of the net distance of the flight path to the actual distance the flight traveled. The closer it is to 1, the straighter the path is. Figure 6 shows an example. Both lines have a net distance of 5, but since the first line is straight, it also has an actual distance of 5, which gives it a linearity index of 1. The second line is curved, so the actual distance is greater than 5, which gives it a linearity index less than 1. A Wilcoxon range sum test determined that EVAR flies had a significantly less straight flight path compared to wild type, as can be seen from figures 5 and 7. Figure 8 compares the maximum height each fly reached, and it can be seen there's also a significant difference between the two types of flies. EVAR flies had difficulty maintaining altitude after takeoff, while wild-type flies can maintain flight. Figure 9 compares the velocities of each fly, and it can be seen that unlike for linearity and maximum height, there is no significant difference between the two groups of flies. These results show that the ablated interneurons play a role in the steering of the fly in flight, but does not inhibit the ability to fly, as there was a clear difference in flight pattern between the two groups of flies. The lower linearity and maximum height of EVAR flies indicates that although they were able to take off, they lacked the ability to stay oriented. Wild-type flies clearly showed the ability to fly with body control, as they were able to fly out of the camera frame in an upward motion, while EVAR flies seem to lack that ability. Figure 4 shows examples of a wild-type fly and an EVAR fly, and it can be seen that both flies have similar takeoffs, but the wild-type flies upwards, while the EVAR drops towards the bottom of the frame. Out of the 20 EVAR trials, only 4 maintained some upward motion before exiting the frame of the camera, while 12 out of the 14 wild-type flies had the ability to fly upwards out of the camera frame. The lack of difference in the velocity between wild-type and EVAR flies, in addition to both flies having the ability to take off and start flight, demonstrates that ablating these neurons do not affect the ability to fly, but rather the steering, which is causing the EVAR flies to fall. The results support the hypothesis that the identified interneurons receive information from the haltere to allow stable flight. Future areas of study would be to compare haltereless flies takeoffs to those of wild type and EVAR in order to further understand the role of these interneurons. Thank you for listening.